Record to the cloud. We're gonna. That should be good. Okay, cool. It's 1:20, so we will get our amphibians and reptiles session started. My name's Anna Case. I'm going to be the moderator for this session. So to kick us off, we have Katie Delaney, and the talk of their presentation is Native Stream Breeding Amphibians in Southern California Affected by Urbanization, Wildfire, and Drought. So take it away.
Thank you very much. Thanks for coming, everyone. Okay, let's jump right in here. I am, let's see, how do I do this? I can get this to advance. There, funny. So I'm from California, and in California, we have lots of iconic national parks. I work for the National Park Service, but my park is a little bit strange. I work at Santa Monica Mountains National Recreation Area in just outside and within the city limits of Los Angeles. So our park is a mosaic, that's our boundary there, and our park is a mosaic of public and private property. A lot of the darker green or the brighter green is public land, so it's publicly owned. Only about 15% of that land is owned by the National Park Service, so it's owned by the federal government. The rest of it is state parks, and there's about 40 other different stakeholders that in various land trusts that run the rest. So some of it's California land, some of it's federal land. A lot of it is private land, so the whole city of Malibu is in our park. And so it makes it an interesting place to study wildlife in an urban setting. Why am I having such a hard time with this this advancing thing? So Santa Monica Mountains National Recreation Area is right on sort of the edge of Los Angeles City. A lot of the hillsides and the parkland looks like this, where it's chaparral ecosystem going lots of deep canyons going right up to the ocean, with some mansions and things dotted throughout. So that's very typical sort of landscape where I'm studying stream breeding amphibians in the area. Our weather pattern is a Mediterranean weather where we get a lot of rain in the winter and zero rain for the rest of the year. And a lot of the landscape looks like this. So there's a lot of open space that's real tiny patches of open space and lots of intact habitat actually. And there's not huge edge effects even with our shrubbery because a lot of it's a shrub ecosystem, but it's literally isolated completely from another patch that could be physically close, but it's isolated with completely impenetrable habitat depending on what kind of animal you are. We also famously have a drought in California, and so this was in 2016 was sort of the height of our drought. We had a big drought between 2012 and 2016. Then this is it from 2019, that year we 2017 the drought broke, but then by the time it was 2019, 2020, and then 22, 23, with the exception of 2021, we've had pretty good rain actually. And one of the things I'm going to talk about is the fire that we had in our area in the fall of 2018. And that's the sort of fire effects part of my talk, where we had the largest fire that had burned in the area ever. So the data that I'm going to be showing you today is a little hand waving. I'm telling my story for you guys, not really any stats here. So I'm telling my story. It's going to be based on this long-term monitoring of stream breeding amphibians. We have a 24-year program now that we've been studying stream breeding amphibians, where we take lots of different data on the stream itself, but we're really interested in the abundance of the larvae of each species that we're studying, and I'll show you some pretty pictures of those guys. So we have a lot of sites throughout the Santa Monica Mountains. So this Santa Monica Mountains is here in the sort of below the 101 freeway, and this is Los Angeles, and that's the San Fernando Valley, and then above there is the Simi Hills. And so we have sample sites throughout the the area, lots of sampling sites throughout the area that we monitor. Some of them we monitor every year and some of them we monitor every 3 years. And we have some partners at Pepperdine and a local resource conservation district in Santa Monica Mountains, which is a state agency that helps us. Some of our streams look like this, which are pretty nice. This is something that if people that live in Los Angeles would be like, where is this? There's nowhere that I've ever been hiking that really looks like this. And most of the time it looks like this. So they're seeing, you can see sort of more natural streams and then our real urbanized streams. And when I talk about urbanization, which I will a little bit more in a minute, urbanization in these streams is not just this kind of channelization, but below this channelization that you see here is a very natural stream with a park around it, like parkland, open parkland. But the effects of the water flowing downhill from this really affects the biodiversity of the stream of the area downstream, as well as this particular area where it's cemented over. So whatever's upstream is really important. So the creatures, Pacific tree frog, this is our most common species in the area that's breeding in the streams. This is a California tree frog that's also another species that's closely related to the Pacific tree frog. This handsome guy is a western toad. And then this is a California newt. So these are the 4 main species that we study. And we're doing a reintroduction project on this guy, the California red-legged frog, which I'll talk about in a second as well. The California red-legged frog is listed as federally threatened under the Endangered Species Act. And so we're doing a reintroduction project, but they're not in very many places. And I have given whole talks about that. You can ask me about that if you want later, so we don't have them in a lot of places, but we do. I don't want to mention that we do have them places. And then we also have all of these things. So these are all of our this is a representative of some of the non-native species that we have that are in our local waterways, non-native fish and the red-eared sliders. We do have a native turtle. We have a native pond turtle. This is a red swamp crayfish that's native to the southern United States. We don't have there was not a crayfish in Southern California. So our amphibians are completely naive to this kind of disruption to the ecosystem, which these crayfish just eat everything. So our urbanization effects, even a 10% urbanization in the watershed. So think about that, just a little, like 10% of the impermeable surface in a whole watershed changes the biodiversity of our stream breeding amphibians. And so what we get is the blue bars over there on the left with the newt and the California tree frog show that they really are not found, they really are only found in our natural streams. So natural meaning less than 10% urbanization in the watershed. And then they're very rarely found in our urban streams. And the opposite is for the Pacific tree frog. They're sort of everywhere. So they're our real common species, which is great. But then you can see also like it's crayfish, and this graph could also be fish. But they're really only found in the urban areas, and they really need that sort of permanent water that urban streams can provide. Toads are just not found in very many places, and so there's not really a huge pattern there. So that's one of the urbanization effects that we discovered early on. Another thing is local extirpations. So we've had a stream that we've been studying for, this will be the 24th year, and we would always get Pacific tree frog tadpoles. Like I said, Pacific tree frogs are pretty common everywhere. They're sort of found in the gross places and the nice places. They did really well this year for some reason, I don't know. But they were sort of uncommon in this stream, and then they just completely blinked out. And we've talked to the homeowners near there, and they don't hear frogs anymore. And so you can see that even heavy urbanization in the watershed and that predation pressure from those non-native amphibians or non-native species can really cause a local extirpation of a really common species. So some of the drought effects that we've seen very little reproduction when there's drought. So there might be water, but there's not enough water. So there might be some water in streams, but you just really get that big sort of gap, that 2012 to 2016, where we really didn't have any reproduction going on with it. Pacific tree frogs, again, this is our common species. This is a really resilient species. It breeds for a long period of time. It doesn't need that much water. It can kind of metamorphose quickly. So they really had a hard time during the drought. And we saw the same pattern sort of happening in 2021 where we had very little rainfall. And that's really pretty obviously because of the lack of water. So this is showing, so 2013, 2014, 2015, 2016, and you really just see that water level going down and down and down. This is just the proportion of dry areas or wet areas in the stream. So it also shows you dry, the lack of water. And so you just have this worsening effect of less and less habitat for them to breed in. Luckily, it's gone all the way back up to 100% since then with the end of the drought, So a little bit about the California red-legged frog reintroduction. This has to do with fire. I'm going to talk about fire for this part. So just as a little background, what we're doing is we're taking partial egg masses from a source population that's nearby. So we just take eggs and then we drive them across the freeway and we put them into these tadpole rearing pens. There's sort of pens in the water there. That's Tupperware's full of eggs that we're going to put in those pens just then. And we grow them up. We grow up the eggs and then they're tadpoles and then we feed them. We check on them a couple times a week and then we release them right there in the stream. And so that's been our project. We had that project going for about 5 years before we had a fire. So this is exactly what sort of it looks like inside the pen. They get hungry and we feed them and and then we release them. So what happens in our area with fire in Southern California and the chaparral ecosystem, it literally burns everything down to the ground to nothing. So there's nothing left. The chaparral burns completely. Unfortunately, with the California red-legged frog population, that red perimeter is our fire perimeter and the red dots are where I've been trying to reintroduce the frogs. And then the source population is up there. So the source population sort of looked like this afterwards. So effects of fire on this project, and then it's also affects, of course, the other species. But this is the source population. We had some direct mortality that was found. This is a before picture. So these are pens in the water there. Notice that rock behind there. So afterwards, it's just completely filled in with debris. That the pens weren't cut in the water at the time, but this is just to show you as an example. But it just fills in with debris. And so these animals, they have these sort of catastrophic events, and if they happen too often, we really can't. So now that this had frogs breeding on its own, but now since the fire 5 years, we don't have any habitat and the frogs are gone. So we're basically starting over, but the habitat sort of still looks like this. So we still have this very negative effects of fire and the debris afterwards that sets back our reintroduction project, and we're just, moving along. So just to conclude, one other fire effect that actually was positive is that Western toads seem to like fire. So they like big open areas, sunny washes. So they like it when the pools get filled in and it gets sandy and it's sort of open. And so we actually started finding toads breeding in lots more places after the fire. So just in conclusion, so the species vary in sensitivity to urbanization. Drought decreases reproduction in our most common species, so probably our other species as well. I have to get to that analysis. California red-legged frogs have lost habitat because of fire, and Western toads enjoy a terrible fire. So they're one thing that may win out. Climate change is our immediate concern. So one of the things that we don't really know what's going to happen in our area is, are we going to have more fire? That seems to be the prediction. Or are we going to have longer and more severe droughts? In Southern California, it's unclear. We may actually get more rain. So this is just this unpredictability that we have to deal with as land managers and as conservation biologists to sort of stay adaptive and keep our sort of heads in the game and flow with the punches here as far as trying to conserve all of these species. So thank you. I have tons of partners for this work, and so I just want to thank you for listening. Thanks.
Plenty of time for questions.
Okay. Yes.
Was there some sort of development that you could point to where threshold was crossed with the extirpation you talked about before and after, like a clear line in the sand?
No, not really. No. So there were tadpoles there and they were rare, they were sort of, but we don't really have an idea. It wasn't like the crayfish increased. So I think what'll be really interesting, and I was just talking to my colleagues over here about it, is we have a graduate student who may be looking at our long-term data and looking at other places where maybe there was a spike in crayfish and we just didn't really note it. and we do have that data, but we haven't teased it apart. I don't think there was a spike in crayfish, but I haven't looked at it specifically, but it could be something like that, that just takes out this small population and never get, the interesting thing about that place is it's a there's Pacific tree frogs upstream. So why aren't they getting washed down in big events and sort of sticking again? And so I'll be interested, we continue this monitoring. I'll be interested to know, like, in 10 years, do we all of a sudden get them popping up again because they've moved back in the area? And of course, that kind of movement, all of you all know, we're studying urban biology. Movement is one of the major problems studying animal movement in urban areas, in urban areas. So, the frogs don't move like they used to. So how they're being hindered from getting to the next canyon is also an interesting thing to think about. Yeah.
What is left of the red-legged frog population that you're working with? Like, how are you moving forward with that?
Angrily. Sadly, no, it's going okay. We have of all those dots on the screen, we have one of the locations that's doing great. They're breeding on their own still. They've been breeding on their own for 7 years now. So they kind of avoided the worst of the fire. The other places, there's a few frogs hanging on here and there, and the habitat's gradually coming back so that we can put the pens back in the water. We have a partner at Santa Barbara Zoo, and they grow up some tadpoles for us every once in a while, and we can throw tadpoles in the water. So we don't need the deep water to do that. A couple of the places just still aren't recovered enough. Some of them are recovered enough that this year we were able to do this process, the eggs to the tadpoles to the pens and release them. We were able to do that in 3 of our 4 streams. And so that, we're getting there, but it's been, 5 years. so we're sort of like starting over in that regard, but all hope is not lost. The source population is doing great. I should say that. The source population burned too, but I think that they just that's why they're still there. So they survived those frequent fires. Yeah. Yes.
Great work.
Thank you.
That image of the fire and the impact it's having on the stream. Curious to know, how is it impacting the connectivity of the stream for them to be able to migrate down? Clearly from that image, it looks like it's completely making it more.
Oh yeah. Yeah, that's a good question. Making it more unconnected, did you say?
Yes.
Yeah. Yeah, that's a good question. The water still flows because it's spring fed. So it didn't look like it in that picture. It just looked like mud and there was no water, but it still will flow. It's like a little braided stream, like just going down through there, where it used to be like pools and it would pool up and it would be a little bit slower. And now it's just rushing through there as a little stream. So it may actually facilitate more movement of things, but they're not able to sort of stick anywhere. They need to sort of be, stop and live. And they'll just like be washed out into the ocean if they keep going. Yeah, all those streams go to the ocean. So yeah. Okay, thanks. Am I done? Yeah.
Yep.
Okay.
I think if anyone else has more questions, then I'm sure you can find Katie during the break. So cool, thank you all. All right, next we have Andre Felton presenting amphibian exposure to microplastic pollution linked to hydroperiod dynamics in an urban IRES system. So.
All right. Sorry. Thank you. So good afternoon. Sorry, I'm making sure I'm on time. So interesting about this project, kind of talk with my hands as well, is that this is the very early start of a new project of information that we didn't expect in a current project. So you're going to kind of hear that story with not as much data as we are about to launch this next study, but really excited about it. So yes, let me go right into it. So yes, amphibian exposure to microplastic pollution linked to the hydroperiod dynamics in an intermittent river ephemeral stream system. If it works, is it? There we go. All right, sorry, it's jumping ahead. All right, so we already know that amphibians are declining worldwide, and it's due to a myriad of impacts. But one of the things that really is catching a lot of attention is the emergence of infectious diseases and new chemical contaminants. Particularly, as you can see here, we talk about rhinovirus, dendropatitis, and a few other infectious diseases that are also decimating populations. But when you look here, what really happens to catch the attention is that microplastics and nanoplastics are one of the most prevalent trigger happy chemical contaminants that we have understudied so far. And it's because of the fact that they are an entire suite of chemical contaminants. They're not just something that can be absorbed or adsorbed. They also have a physical presence. So you have that compound impact of them being able to absorb things, leach it back, as well as then their physical parameters and their physiochemical properties that essentially make any issue far more complicated. And so one of the things that the study we were looking at was already understanding microplastics in urban systems, but in these intermittent and ephemeral streams, amphibians that are native to San Antonio really thrive in these habitats. And we started to come around to understand that they have unique exposure routes and vulnerabilities throughout their development and phenology that started to show up in the last few years. And so just to give you a little more background about microplastics as to why that is so important, is because of the fact that plastic production is going to continuously increase. It's exponentially increasing each year. Pretty much as of 2019, we had 368 million metric tons, and that is going to thoroughly increase, driven largely by single-use plastics. Which again are typically what we find in these streams that they pool. And though this isn't a talk about that, you'll see how that impacts amphibian reproductive biology. But these ephemeral systems, which is where I really want to focus, is that they are designed to essentially use the hydrology and the hydro period of these pools where they are connected, and then periods where they're not connected to move things through. Typically, if you have an urban stream, there's a lot of pollution. And what we start to find out is that plastic pollution seems to mirror, and therefore subsequent exposure seems to mirror these hydroperiod dynamics to which amphibians, particularly the frogs and toads that use these pools, breed exactly at the time that pollution is highest. And that becomes a very interesting story that we did not expect to see. And so, as you can tell here, there's a paucity of research looking at this because we understand amphibian biology and physiology and physiological responses in general lotic systems and lentic systems. But what we're getting here now is that a system which is the dominant aquatic freshwater river system, which are IRES, very little research has been looked at to see how amphibians are dealing with these new emerging threats in these systems here. So the original study was just to understand the spatial temporal distribution of microplastics in these systems. But through the data process, or through data collection, we started to understand that amphibian diversity was starting to really show an interesting pattern with these streams. So we tacked that on, to which some of the undergrads that are here are going to end up doing the study that carries on from that. So I'm really excited that they'll be taking this and running with it. Furthermore, we then started to evaluate how microplastic concentrations, how microplastic concentrations seem to influence or co-occur with amphibian breeding in these pools, especially in urban systems. And then last but not least, we wanted to assess how tadpole vulnerability and exposure essentially is impacted by these parallel relationships we started to see, or parallel patterns, I should say, of microplastic concentration with hydroperiod dynamics in that. Okay, so our system here is in Texas, as you see here, with Bexar County. And in Bexar County, where San Antonio resides, we have two major urban watersheds. On the left, you can see that we have Leon Creek, and then on the right, we have Salado Creek. They both basically mirror the west and eastern portions of the city running north to south. As you can see, the blue line is the main stem. There are several tributaries that feed in, but each blue solid line on each side is the main stem, to which we sampled and monitored for 3 years. So just to give you a little understanding of our sampling system here, again, we monitored each one. We had 13 ephemeral pools and 13 runs. That means between each river, because they are IRES systems, there is a significant reach that are defined by levels of intermittency, levels of ephemerality, and then as you move further down, completely running like perennial most of the time. So it was also a great opportunity, since we found amphibians at every site that we had, to compare what was their exposure route and what other impacts they would have given where they're found. And of course, connectivity matters, but because of the fact that larger sections are ephemeral, they are more times than not disconnected. So we get to see kind of some interesting patterns that could potentially show up, I should say, not definitive. right? So just to kind of give you a little more understanding here of these pools, if you don't know the ephemeral systems of the Southwest, what you can see is that for Leon on the left, Salado on the right, you have two columns. The left column on each side is when these pools are connected and full of water, right? And they can hold water anywhere from as long as up to 6 months. We've tracked these for years. We actually have roughly about 10 years of data. My study was for 3 years so far. So we know that they can hold water up to that, but they're highly variable. And then what you see on the right side is the same site. Some of them only a week later, some of them a few months later, but you can see how that ephemerality going from the top and then some of the perennial reaches as we get to the bottom, such as there. And you can see that hydro period really does impact what could happen there. All right, just to say again, sampling what we did, you can see also one of my undergrads here who's doing all the work while I get to sit there and just take pictures, is that we took water samples at every site. We took sediment samples, and then we started to actually collect information on the amphibians we saw there, but we wouldn't do it at the same time. We would come back that night. Okay, and typically take that information just again, not to disrupt or confound anything when we're collecting samples and things of that. I always like to talk about pretty much how we process microplastics, since that's at the early forefront, and you can see this process of getting microplastics out because that's where it started. We took a 12-month sampling where we were out every other week. Sometimes it felt like every other day. And then we subfractionated it. From there, after extraction, we isolated every sample, water and sediment, to see how much microplastic content was in each volumetric collection. And then we identified it. But what's better is to show the people who did the hard work. Right. And you notice I'm not in any of those pictures. So you see here again, series of undergrads and my son. Yes, I do make my son do this work as well. That pretty much shows how this work is. But here's where it gets interesting. I'm jumping right into the data. So here, really quick, just to show you, this is microplastic concentration. This is the average microplastic concentration in the surface water alone. All right. What you see, the color code from teal to purple to blue lets you know the independent reaches. So we have upper reach, which is the most headwater in teal. We then have purple, which are our middle reach sites. And then here in the blue, we have our lower reach sites. That also correlates to what was ephemeral, intermittent, and perennial. So the color code kind of lets you see that here. Also, the letters let you know for significant difference. And what's interesting is, of course, most of the perennial had higher levels. They're perennial. They keep running all year round. When you're out there every other week, you're going to see that. But for our ephemeral and intermittent, you saw levels of difference, right? Due to the pool size, depth, when it was wet, when it wasn't, other factors in that. And why I'm going to focus on that is that's where our amphibians mostly were. We did find some here, but they were typically washed down from that. Okay. And then before I jump into that, I also wanted to look at it at reach and see if maybe the reach had an issue. Why does that matter? Because the upper reach and the middle reach both are highly urbanized in these watersheds. The lower reach is not. It's more agricultural. So we wouldn't expect to see as much as we saw there, but that's more than likely washed down from the upper streams. But you can see that for microplastic content and other variables that influenced it, the middle and the upper reaches were not impacted. So that lets us know that whatever we may have seen or will see in the amphibians may be a different variable than what impacts the microplastic concentrations, even though it overlaps with their vulnerability to these patterns we're seeing. All right, now why I'm showing you this is again the box plot that shows microplastic concentration in the sediment and the water. Okay, so the 0 is a neutral. That's just to represent above ground and below ground. Okay, so beneath you can see that clearly there's a lot of microplastics given in our interquartile range, but the max is at the extent of everyone. Thank you. And why that matters here is that each one of these outliers is when we collected things within 72 to 96 hours after a rain. Each of these outliers is where we found basically larvae, larger tadpoles, metamorphs. We found them all during these times. Right? We did not see them when it was essentially leveled out or longer than that period. They've pretty much already started metamorphosizing, going on forward. But it's interesting that during these times is where we saw it. Why does that matter? It's because majority of our microplastics were fibers that were attached to biofilms, fibers that were attached to periphyton, many of the things that they feed on at that time. And why that also matters that many of the tadpoles, which we had 6 different species, some overwinter. So the fact that they were in the sediment for that time when it was not wet. And then when it got wet again means that they had differential vertical exposure as well as then temporal lags to what was immediately washed in and what had been in the water or it settled at the sediment from the last precipitation event, which could have a legacy effect that we just don't know yet as we want to look into it. All right. But then from there, when we see fibers were predominant, we also see that we had a myriad of different types of polymers. That means you have different impacts that could happen given the chemical basis of each core polymer and the copolymers that are added to them. Right. Just to jump ahead, why I also want to say that is because we did do this for a year. Only during these times did we see tadpoles and all of the actual amphibians during each block. Following that time is when we started to get water again. And we didn't see that. So during those blocks is when the drought hit San Antonio, yet we still saw them. Follow that, we didn't see them. Their phenology was completely thrown off at that point, which was a very interesting point until this year where we start to see them again. But an undergrad is going to be doing that work, so I'm not going to bust that bubble. Again, we looked at it reach and we saw similar things following the concentrations there. Only for time, I'm jumping ahead. Thank you for that. And so overall, the results show, I'm going to jump past the results for microplastics. It's just that we have a ton of microplastic that's lower than most perennial systems, which makes sense. We have a highly urbanized, but still residential input to our streams in these greenways, right? But what becomes more of an interesting story is that where microplastics are already present, we may actually have a correlation where amphibians that are results from breeding events during that following precipitation events in that 96 hours, what we see is that they are having chronic exposure from the start because microplastic concentrations are the highest at that point from resuspension and recent input. So you have that as they are breeding and then being laid, they are at their more than likely their most vulnerable, but then you have ones reemerging from overwintering that now have a stratified impact from what was settled into the sediment, as well as they move back up and down through the vertical stream, which means they also have differential impacts based on the morphotype and the polymer. So these are questions we are definitely interested in looking forward to, which they are going to look at, which I'm excited for. I'm sorry, we don't have actual data just on that just yet, but I had to present the story because it was super interesting to jump into. I'm going to stop for time and anything else. Oh, these are future steps and we are done. And these are all the teams that I would like to thank for helping with this. You can see our lab at the center. You can see several of us on different trips. And of course, my children, they have no choice but to continuously help with this study. Thank you for all the funding sources that have continued. Thank you. Should I stop?
Yeah, you're good. We have time for questions.
Okay.
We have plenty of time for questions.
Okay, awesome.
Okay.
Then I'll go back. Yes, ma'am.
What do you think that the effects, or do people know what the effects of microplastics in tadpoles is going to be? Like what biologically is going to?
There's a few studies. There's only been a few studies that have looked at it. You have everything from increased mortality, depending on the type, all the way to just increased oxidative stress. Typically, it depends on the size and the morphotype. So whether it's a sphere like a bead, a fragment that's oblong and irregular, a film which is very thin, a line, all of those things can have varying effects, but it seems to go to every species kind of responds a little differently, as well as then increased egestion rates dependent on route of exposure. But most studies have not looked at amphibians yet. We should have some work coming out to show more about what's happening. Just this past month, there was one study looking at metamorphic timing, and you saw that there was a delay in xenopus, but that delay was not shown to be significant. But at the point of the delay, metamorphic climax was they were essentially wider and a little bit heavier, but you didn't see anything else save for acute responses.
What about adults?
There hasn't been anything done on that so far as I know so far. I'm looking at. Great question. Great. We do have a study that will be designed, so we'll be doing that in 2 weeks. That will start it off chronic exposure to see all the way what happens, looking at morphometrics, microbiome dynamics, translocation. So we are excited to see what happens in that as well. Yes.
Did you come across any papers related to plastics?
No, so I'm so glad you said that. Cook sucks. No, I'm joking. I'm joking. I actually wanted to do work looking at that, but no one has reported anything. If anyone's looked at it, no one has reported it yet. But I do think that's a very interesting question. A way around that. is looking at the microbiome community and seeing if there's any pattern of impact from microplastic exposure on that, and using it as an index or proxy for potential impacts for vulnerability or susceptibility to chytrid, but nothing directly yet. Yes.
So you hinted at a legacy effect. And I was curious if you know of anyone like working on that in terms of like looking in vitro at gene expression.
We do have a doctoral student that just joined who has that expertise from his master's. So that is a planned future study. To answer your question directly, though, currently I have not seen anything, and I'm not aware of anyone doing that yet that has reported it. I believe there may be a lab. I believe there may be a lab. I think James Madison in Wisconsin might be doing something to that, but I could be wrong. But I know that we will be looking at that also. So I thought that I saw a hand over here. Thank you.
Nice.
We have a few minutes. Yeah, so, two, there we are. Okay, so I'm gonna share my screen again and just do it that way. Okay, so this computer just has a slight slag. Has it?
I forgot to look my talking.
Oh, nice. Okay, yeah, so we're just gonna leave this right here and we'll do it right before. It'll be fine. Thank you so much. Yes, so it does. It has just a little bit of a lag, so just know that. Like it, yeah, it might take it a minute. Mhm. Yeah, don't do that because it'll just freeze. Okay, let's see. Make sure everything. Oh, perfect. Did I do it?
So next, we'll start with this really. Next, we have Elizabeth Hauk. Okay.
It's a weird
Elizabeth Hauk. And they're going to present on the reproduction in relation to urban level in an invasive Florida lizard species. So.
All right, awesome. Hi, guys. I'm here to talk about a project that I did for my graduate thesis down in USF St. Pete, so down in Florida, titled Reproduction in Relation to Urban Level and Invasive Florida Lizard Species, or Anolis gray, the brown anole. And if you guys went down there, you're more than familiar with these guys. Okay, so the big question, the overarching thing that I wanted to look at was how do brown anole reproductive parameters vary across urban level? Again, they're everywhere down in Florida. They're a really easy species to study this, so that's what we went with. A lot of you in here probably know this, but urbanization is kind of a broad topic. There's a lot of different stressors that urban environments can introduce to reptiles, things like heavy metals, pesticides, organochlorides, PCBs, increased stress levels can all happen and occur within reptiles in these urban environments. A lot of these are known EDCs, or endocrine disrupting contaminants, which is basically anything that is capable of disrupting the reproductive or endocrine system within these animals. And they usually are in higher concentrations within urban levels as opposed to these rural areas. So things like, again, heavy metals, PCBs tend to really concentrate in these areas of urbanization. It's an issue because, as we all know, urbanization is increasing pretty rapidly across the world, and therefore these pools of these EDCs and these urban stressors are also increasing in these areas. So this is just a quick table, some background information of stuff that we do know on this topic. It is not necessarily widely studied, but different areas of urbanization. So vehicular runoff is a big one. It's a big one that's known to put some of these EDCs into the environment, particularly heavy metals. Damming, general urbanization, so increase of population density in people, buildup of highways, urban environments, stuff like that, are all known to, in some shape or form, negatively impact reptiles. So things like increased parasite load, mortality, decreased abundance have been shown up in these urban species. So if we're going specifically between urbanization and lizard reproduction, again, this is not something that is widely studied. but for a few of the studies that have shown up on these, again, is usually concentrating more towards the heavy metal or PCB side. But these have been shown to have really adverse effects on lizard reproduction. So everything from increased mortality rates in hatchlings and embryos to really, really awful malformations. Now, these are in really high concentrations of known indices, like cadmium is a big one that has been studied. It's shown to decrease the development of eyes. Skulls don't form correctly with these embryos, the hatchlings. It's kind of a mess. But again, we don't really know a lot outside of that. So lizards are pretty interesting because they are uniquely susceptible to contaminants like this in urban environments. They are, for most of their species, and for the anoles, are mesopredators. So kind of a mid-length predator, not really an apex predator, but they can have the effects of bioaccumulation up the food chain when they are eating all of their insects or even sometimes smaller reptiles. So they can get these contaminants up through the food chain. It does bioaccumulate. So the higher these animals up are in the food chain, the more susceptible they are to these toxic contaminants. Obviously, they have lungs. They can breathe in contaminants from urban environments through lung exposure. And they can also absorb it through egg deposition. They have very porous eggs. They're really, really tiny when they're first laid, but they will double in size by the time they are laid to when they actually hatch. They are not born with all the moisture in them that they need. So they need to continuously facilitate gas exchange, and they need really damp environments. So anything that's in the soil that could potentially harm these hatchlings, these guys are going to be exposed to. So why them for these particular studies? Outside of the fact that they are useful bioindicator species because of their unique susceptibility to different contaminants and urban stressors, they are easy to reproduce in a lab, especially when we're talking about the brown anole. There's a lot of precedent for them being used as a lab species because they are, again, so abundant. In Florida, they are the most abundant wild vertebrate species. So they are everywhere, despite the fact that they're an invasive species. They have pretty fast generation times. So within the span of a few months, I was able to get a lot of data on the growth of their hatchlings, which is not always easy to get in other vertebrate species. So with that being said, there's still a lot of knowledge gaps when we're talking about this topic. So discernible trends and reproductive parameters in most vertebrate taxa in areas of moderate urbanization are very unclear. So when these studies are done in urban areas looking at reproductive impacts, it's usually a very known, very high, very polluted area, like a mining site or a phosphorus plant that they're being taken from. And then the comparative site is usually somewhere out in the middle of nowhere, a national park, something that's very protected. So it's two kind of very widely discernible areas. So, very rural, very urban, or just very urban. Anything in the middle is not very well studied. And the trends that we do see kind of vary from paper to paper. Sometimes effects of urbanization increase in moderate areas, sometimes they go down. A discernible trend is very hard to find in existing literature. On top of that, when we're looking at reptiles specifically, avian species, so birds, are used as surrogate species for reptiles. Birds and reptiles are very closely related. I know everybody in here knows that. However, this is not very helpful when we're looking about specific reproductive outputs, reproductive measures, the way that they reproduce, the way these hatchlings are impacted, that's really not accurate. Like I just said, a lot of these reptiles have very soft porous eggs. A lot of them are being laid in the ground. That doesn't happen with most bird species. They have harder eggs. A lot of them, especially the ones that are used in these studies, tend to be songbirds or birds of prey. They tend to be up off the ground. It's just really not accurate with how they're being exposed to these potential stressors. So that leaves a lot of knowledge gaps when we're talking about reptiles in general. And most importantly, you actually kind of touched on this a little bit, existing literature when we're looking at reproduction does not usually focus on multiple generations. They will look at adults, contamination within the adults. They might look at reproductive output, so how many eggs or hatchlings are hatched. But when we're talking about the physiological impacts of a second or third generation, there's really not that much existing literature out there. So that brings us to the hypothesis that I formed. So the reproductive parameters that I wanted to look at were hatchling survivorship, hatchling initial size, so how big they were when they first hatched, and hatchling growth rates. So it makes sense. You would think you would see a pretty clear line with the low, moderate, high, with these reproductive parameters decreasing. So hatchling survivorship will decrease, hatchling size, growth rates will decrease as urbanization level increases. So there were kind of two parts to my field methods here. part one took place out in the field. I had to go get these suckers. So I broke up my urban level, make it sweet and easy, high, moderate, urban, 3 from each. I use what's known as the HFI, or the Human Footprint Index, to discern the urban level of each of my sites. So this is basically a very large collection of information that takes in a lot of multiple factors of urban and human impact on different spaces. So built up environments, so that's any urban structure, buildings, highways, human population density, artificial waterways, air pollution, light pollution, a whole bunch of different factors, and they put it all into one graph. And then I determine my gradient that way. So 0 to 50 is kind of the line that the limit that this HFI has. 0 is least impact from humans. 50 is the most impact that a human could have, which is basically as far away from its natural threshold, its natural base as possible. So my field sites went from 5 to 38. The lowest ends for the low urban were around 5 to 12, and then the higher ones went up to 38. 38 was the highest. So I captured all these lizards using the noosing technique. I don't know if you guys have ever seen that, but it basically involved me walking around all summer with a fishing pole with a little line at the end and scooping up these lizards. But we wanted to get 4 breeding pairs from each field site. So 4 male and 4 female were taken from each of these field sites, and they were kept together as a breeding pair. They were then brought back to our lab where each breeding pair was kept together. So their mating season and their breeding season usually goes from late spring, early summer. So for the duration of that, these breeding pairs were kept together. Any eggs that were recovered, which we checked for them every single day, was immediately incubated. Again, they are very susceptible to desiccation. They need, it's a lot of work. They need to be kept in a very, very moist environment. Again, these eggs need to double in size before they're ready to hatch. So each egg was taken out, incubated separately, and then when the hatchlings did finally hatch, all siblings were housed together in these terrariums. Okay, so once the hatchlings finally hatched, they were measured immediately. So this is an initial SVL, snout to vent length, that I used for my data that was taken. Mass was taken, but the variation was so little that it really didn't give me much. They were marked to be able to tell them apart, and then a repeat SVL measurement for every single hatchling was taken at the beginning of every single week. So that's how we got our growth rate. Any mortality was recorded immediately. Again, these guys were checked on for multiple hours every single day. So it was pretty accurate from what we got. Going to our results. So for the most part, they were pretty clear. So hatchling survivorship did decrease pretty significantly in low urban environments. We had a 60% survivorship rate in high urban environments, but 100% in low and moderate. Now this trend was seen for all the parameters. The same thing with hatchling body size. High urban environments were much smaller than low and moderate. There wasn't a difference between the low and moderate, nothing significant, but they decreased significantly in the high urban environments. Same thing with growth rates. You guys are seeing the pattern here. Just a quick throw in, I did take measurements for the adults. There was no significant variation in the adults, neither the males or the females. I did focus on the mothers because you can't always prove that the lizards in the tanks were the fathers of these hatchlings. So it's not super accurate data, but I do like to say that there was no significant variation in size of the male or female adults. With the hatchlings, it was pretty stark. So I'm just going to skip this for time. Basically, the takeaway here was there was significant difference between high urban areas and low and moderate on all of the reproductive parameters, but there was little to no variation in the low or the moderate areas. Something that I do want to touch on really quickly is the possibility of maternal transference or maternal offloading. So this is something that is not particularly well studied, but it is kind of an up-and-coming theory when we're talking about maybe impacts that we see in hatchlings or young or embryos, and we don't see any physical or apparent malformations or issues with adults. This is basically a process in which mothers can offload these toxins within their body through their embryos, through eggs, eggshells, things like that. Again, these toxins, EDCs, tend to accumulate within tissue. They are very hard to shed. That's why they accumulate up the ecosystem or up the food chain. This might be why we're seeing results in hatchlings and it's so stark and so apparent, but the adults appear perfectly fine. Again, this is very understudied. The only species that I see a lot of studies with this are elasmophorans, so sharks, rays, skates, and turtles. There's some really interesting data that's being put out. It's very apparent with how these embryos, these hatchlings, these young are being impacted by this, but not the adults. And that makes monitoring the health of these species in these urban environments very difficult, because in order to really get the impacts, you need a multi-generational study. These adults might seem perfectly healthy, no decrease in body conditioning or anything like that, but then their offspring, those embryos are where we're seeing the impacts. So it's hard to just, see these effects as is. It also shows that these high urban areas should possibly be where we want to concentrate our efforts to really monitor the health of these species and to actually understand what the physiological impacts of these contaminants with increasing urbanization rates are. So again, just a quick wrap up here from what I learned in the study. Urban level does have significant impact on brown and anole reproduction, specifically when we're talking about high urban areas. So these results suggest that hatchlings may be disproportionately impacted by environmental contamination. Again, there was nothing wrong with the adult females. They were all perfectly healthy. And yet some of the hatchlings from high urban environments just kind of dropped dead. They were much smaller. They were not as healthy, but we didn't see that in the adults at all. So future studies are obviously required to be able to kind of disentangle the various effects of urbanization. Like I said, there are multiple stressors and multiple contaminants that are put into the environment when we're talking about high urban areas. And it can be difficult to pinpoint which one or multiple ones might be causing these results, but that is definitely something to look at in the future. And again, there are still a lot of knowledge gaps that we have when we're talking about urbanization, EDCs, and reptile reproduction. I think this is an interesting way to kind of start to look at this problem, but there are a multitude of other species, environments, contaminants that we should probably look at and prioritize when we want to look at the health of these urban animals. So just a quick acknowledgement to my advisor and my committee at USF for all their help with this project. And that's it.
Time for questions.
Yeah.
I'm curious how the juvenile survivorship issue would feed up to population, stable population sizes in the different contexts. So I don't, maybe there's not data for this, but for example, I can imagine that there would be more predation due to predation. So sorry, more death in juveniles with predation in the less urban areas, so it actually have compensating effects.
Yeah.
Do you happen to know if there's like data on the stable population size?
When we're talking about brown anoles, not necessarily. They're a pretty invasive species, and unfortunately, people don't tend to care about them as much in their survivorship. I think that's probably an interesting point, and I think, as far as I know, I don't know any actual studies that are looking at that. But I just know it's in the lab where everything was very controlled and there was no outside force. The mortality rate was pretty stark, with no other factors that were potentially determining that. But yeah, I don't know specifically with these guys.
Very nice talk.
thank you.
I was just wondering, in these field sites, these populations, I mean, you're kind of got them from the same kind of area, but are they like hyper isolated? This is an invasive species. Like, do you think there could be other effects outside of sort of contamination, such as genetic effects? If they're massively inbred to some degree, then you could have this sort of filter that occurs when they're trying to lay the egg, some sort of issue there, some genetic incompatibility from this type of inbreeding. But maybe, there's so many, you're like, this is a problem.
That's always a possibility. I can, for sure say no. But they are, like I said, they're extremely prolific, and they have been coming up through Florida for a very long time. They are very outspread throughout the state, as far as I know, since Florida is kind of where I'm concentrating on these guys from. Like I said, I can't for sure say that that's not a possibility, but I do think they've established themselves and they've widespread enough that might not be the most pressing issue. But I don't know of any studies looking at it. So.
Are you testing the eggshells at all for just any like contaminants?
So I did actually, that's part of this that I cut out for time, but I did look at heavy metal contamination within these sites. I looked at it through the 9 field sites. The soil contamination was pretty stark. It was pretty insane how high it was in the urban areas. I did do just a couple samples of the adult tissue for urban or for high, low and moderate urban. There was no variation in pigment accumulation in the adults, but in the hatchlings, I only had enough funding to look at 6 of them, but it was higher in the hatchlings than it was in the adults, which is pretty insane considering the size difference. And this was from whole body tissue, so this wasn't isolating anything. So not, among urban levels, but between the hatchlings and the mothers, it was a pretty alarming difference. Yeah, but I did look at that. I think looking at the eggshells though is probably the next step. I still have them. I'm just looking for the funding to get that testing done. Yeah, exactly. It's expensive.
Yeah.
Tissue sampling is really expensive. Yeah.
I was just curious, like, there's so many directions that you can go when you're talking about components like ecotoxicology, but I was wondering, like, if you plan on looking at coupling components and if like climate stress too, that's also influencing like the survival threshold.
Oh yeah, I think that would be a great way to look at it. I don't think the brown anoles are necessarily the best study species for that, just because they're doing so well in urban environments and they're really thriving in hotter climates. But there, like you said, there's so many ways that you can take this depending on the species that you pick, that I think that's, again, it's convoluted when you're talking about the stressors of urbanization and, human impacts. I think that could absolutely be causing issues like that. Yeah. All right, awesome. Thank you, guys.
We're a few minutes ahead of schedule, so does anyone have any good reptile jokes?
Yeah, let's try and keep it on schedule if we can. Take a minute or two.
I did a whole PhD on turtles and I don't think I even have a good turtle joke, so. Any alligator jokes? No.
I think so.
This is my phone, please. Any urban wildlife jokes that anyone has? We'll broaden it. Yes, no? Yeah, I guess we will stare awkward. You are the second to last one, so one more after you. I guess I did see a pretty good one about a good dad joke about alligators, where I was gonna make alligator for dinner one night, but I only had a crock pot. Good, I got the right spot.
All right, let's do it.
All right.
Let's do it.
We won't stand here painfully anymore. All right, so up next is Adam Rosenblatt presenting how are alligators affected by living in an urban environment.
Thanks for that filler. I appreciate it. Yeah, thanks for coming today. I'm going to be talking about some of our recent research on alligators living in Jacksonville, Florida. And the motivation for this work is obvious to everybody in this room. Urban areas are growing. We all know that. And nowhere is this more true than in the Southeast. According to this study from a few years ago, urban areas in the Southeast are predicted to at least double in size, and at a minimum, and then at a maximum triple in size by 2060. And this map here shows all the red areas, or the areas that have the highest likelihood of becoming urbanized in the future. And that's going to be impacting the wildlife in the Southeast. Now, the other thing we have going for us, from my perspective as a positive in the Southeast, is that we have recovering alligator populations. Alligators were, hunted to a very low population level through the first half of the 20th century, but then they're probably one of the best conservation success stories in this country that a lot of people don't know about. But their populations have rebounded really dramatically over the past 50 years. To the point where in Florida, you know, we have about 1.3 million alligators. In Louisiana, there's more than 2 million alligators. So the populations are coming back, and they're pretty robust. And what this means is that there's going to be a greater likelihood for human alligator conflict in the future, and we should be trying to understand how alligator populations are impacted by these growing urban areas. And it's a really unique opportunity to study alligators in urban areas, because in most of the world, wherever you have urbanization, the large predators are the first ones to be pushed out, right? Large predators usually need lots of space, and humans don't tolerate them that well, so they get pushed out very quickly. But in lots of parts of the Southeast, we still have cities with these relatively large predators, so it's kind of a cool opportunity. So I'm going to tell you about two studies that we did. One is ongoing, one's completed. And this is Jacksonville, where University of North Florida is located, my university. It's the largest city by area in the entire United States. So we have a gradient. We have areas that have, you know, high human density. We have areas that have very low human density. Because there's only about 1 million people live in Jacksonville, but it's massive. I mean, it's bigger than, you know, Houston or Phoenix or anywhere else. And so we wanted to ask a couple questions because also, I should point out, Jacksonville has the largest river in Florida that goes right through downtown, right? That's the St. Johns River right there. And historically, this would be ideal habitat for alligators. So are they still there, or have they been pushed out over time? So the questions we're interested in looking at are, how do alligators distribute themselves in an urban environment? And then what size classes are even still remaining in a place like Jacksonville? So the first study we did, we were looking at tributaries of the St. Johns River, outlined here in red. We're looking at 9 different tributaries from the south through the north. And we wanted to see, could we figure out answers to the questions that I just posed? Just to show you the sort of differences in these kinds of tributaries, Black Creek on the upper left is a tributary that is not very impacted by urbanization. It's a protected preserve. And so there's very few houses. There's not a lot of industry along the, you know, banks of Black Creek. But further down towards the right, we have Arlington River, which runs right through one of the most heavily populated parts of Jacksonville. And you can see by the, I don't have a legend on here, but you can see by the color coordination, there's a lot more different kinds of land use along the banks of Arlington River. There's a lot of houses. There are big apartment buildings. So it's a much more heavily impacted site. So across these 9 tributaries, we have a gradient of urbanization that we're able to sample. So what we did is we surveyed each tributary once per season for 4 different seasons. This is all during 2019. Each tributary, there was an 8-kilometer long transect that we would drive with our boat at a consistent speed. We had 2 spotlights, 2 observers, and a driver on each boat. And every time we saw an alligator, we would record the habitat type that we saw that alligator in and what kinds of habitat were around there. This is a video of what it looks like. If you've never gone alligator hunting, we go out at a boat at night. We have our spotlights, and the video is not playing. Oh, there are spotlights and it's rushing through. What we're looking for, just went right through, okay. You missed it at the end there, but we're looking for these red eye shine, right? If you shine spotlights at an alligator's eyes, they shine back at you bright red, and that's why we do our research at night. These are the results. We published them in 2020 with my grad student, Eli Beal. And we, you know, looked at all the numbers across all these different tributaries, and we had a bunch of factors we were looking at, like the saltiness of the water, like the amount of urbanization around each of the tributaries, you know, light pollution. There were a bunch of different factors that we were considering. And we used multiple linear regression to look at which one sort of shook out as the most important. And by far, the only one that was important was the salinity of the water. So here you can see it's salty as you go up and it's fresh as you go down on this table. And where it's salty, alligators avoid those areas, and where it's fresh, alligators are more abundant in those areas. And that's not shocking at all. We know that alligators avoid salty water in general because they don't have salt glands, so they can't deal with the excess salt in their body. But what we found is that across all these tributaries, there's not really an impact of urbanization directly. So that's what I just said. Gator distribution is primarily limited by salinity in Jacksonville across all the tributaries at a coarse scale. But when we dig down within tributaries, we actually found through a different type of analysis that gators are attracted to vegetated shorelines, and they are repelled by human structures, right? So these are docks, people's docks in the backs of their houses that are pointing out into the water, or if there's a hardened shoreline where the shore has been, you know, reinforced with concrete or a bulkhead or whatever it might be. So at a gross scale, they're not responding to urbanization, but at a fine scale, it seems that they are responding to human structures. The big question we had at the end of this study, though, was where are the big gators? We saw 93 alligators, and only one of them was an adult, and it was a small adult at that. It was like maybe 7 feet long, which is a pretty small adult. So we know, adults have to be there because there are lots of babies around. They must be coming from somewhere, but the alligators clearly are not making themselves known. The adults are not making themselves known. So that was a big remaining question for us. So we decided to do a second study to dig further down on this problem, where, obviously the St. Johns River is a place where alligators might like to live, but also in Jacksonville, there are lots of retention ponds, right? Retention ponds are stormwater, management areas trying to limit flooding. And there's just, I don't know how many there are in Jacksonville, but there's probably thousands of retention ponds across Jacksonville, and they could be alligator habitat. But as far as I know, nobody's ever looked to see sort of what the distribution of these animals is across those types of habitats. Our hypothesis for the retention ponds is that their distribution would be related to how far they are from Guana Lake. Guana Lake, outlined here in red, is the largest known population of alligators in the Jacksonville area. It's just south of the main part of Jacksonville. It's a protected area. It's a National Estuarine Research Reserve, or it's part of one of those reserves. And it's estimated there are about 700 alligators in Guana Lake, right, including some large breeding adults. So our hypothesis was that we would be more likely to find alligators closer to Guana Lake in retention ponds closer to the lake relative to farther away. This is what some of these retention ponds look like. These are ones that we actually studied. The one on the left is actually on the UNF campus, and it is a retention pond that you could see an alligator living in. It has some habitat characteristics that they would like. It has vegetation on the surface of the water. It has a natural forest fragment along one shoreline. And so you could, and also you can't see it here, but this forest fragment is connected to a swamp that's further back, right? So there's some habitat connectivity for them. So it wouldn't be shocking to find one in a retention pond like that. Whereas this one on the right is actually pretty close to my house. And it is, completely manicured shoreline. It's all lawn. There is no forest fragment around. All of these are, trees that were planted there. It's surrounded on all sides by roads, and there are, shops. This is a shopping district that's right around this pond. So, and there's a hardened shoreline in multiple spots around where the lawn has been turned into concrete. So that is an area that we would not expect to find alligators in. So what we did is we surveyed 71 retention ponds during July of last year, and we surveyed each pond twice with 2 spotlights again on back-to-back nights because we were doing an occupancy analysis. And this is a map of what we found. All of the dots are where we surveyed, and all the green dots are where we found alligators. And so you can already see sort of what the results are that I'm going to show you over the next couple slides. But what we found is that only 9 of the ponds were occupied at that's a 12.7% occupancy rate. We only spotted a total of 13 gators, and again, all are juveniles and subadults. Still no adults anywhere in these ponds that we can locate. To dig deeper into this, we did an occupancy model, a habitat occupancy analysis, where we can both model the detection probability of alligators in these ponds as well as the occupancy probability. We looked at a bunch of different factors. So, in some of these ponds, there are fountains. There is, as I said, surface vegetation. These are things that could limit our ability to detect the alligators in the first place. So those are our covariates for detection. And then for occupancy, we have distance to Guana Lake. We have, is there hardening around the pond? Is there fencing around the pond? All these things that could, stop alligators from getting there in the first place. When we look at just the detection covariates, only one of them was significant and was part of the best model as ranked here by AIC, and that was forest. If there was forest on the shores, that decreased the probability of detecting an alligator in the first place, which makes sense because they like to hide, in sort of forest canopy cover. So then we use that for the occupancy modeling, where we're looking at the occupancy covariates, and again, ranking by AIC, these various model combinations. We looked at a bunch of different models. These are just the ones that were the highest performing according to AIC. And the only one by far, again, that matters is distance to Guana Lake, right? That was the only thing that really impacted where we would find alligators. So our summary here for this part was that alligators are not dispersing far from the source populations in Jacksonville. I think that's pretty clear. And dispersal is likely limited by urban land use, right? I mean, what alligator wants to cross a 4-lane road and then, go through a shopping district to get to a pond that is cut off from lots of other ponds, right? It doesn't really make sense to pursue that strategy. The other thing is that larger alligators are removed, right? I mean, we know this. There is a program in Florida called SNAP. It's the state nuisance alligator program, and anybody in the state can call a state hotline and say, there's an alligator in this area and I want it removed, and the state will send out a licensed trapper and they will do that. And we just had an example of this in Jacksonville on the north side, where that green dot is a middle school, and this is just 2 weeks ago. A 10-foot male alligator, this is a breeding alligator, was found in the parking lot of this middle school. And so, obviously this is a safety concern because there are children around. And so a trapper was called and removed that alligator in a very dramatic fashion that made the local news. And it was killed because that is that's sort of the standard operating procedure. There have been attempts to move alligators like this to other areas, but they can make it back to where they were removed from. And so that's not a long-term solution. So if an alligator is longer than 4 feet, they tend to just be killed, and that's just how it goes. So this alligator here, there's a creek called Moncrief Creek that runs right behind where the school is. Right there, it's a big open field, and that connects to Trout River, and Trout River connects to the St. Johns River, right? So this 10-foot alligator was probably living in Trout River and Moncrief Creek for decades. And then for whatever reason, it decided to leave one day and go into a, more heavily populated area, and it lost its life as a result, right? So clearly there's a strong selection against alligators moving far from the source population and going into urban areas. Bold alligators are not rewarded, right, in urban areas. So to get back to the original question, how are alligators affected by living in an urban environment? We know now that they tend to avoid human structures. We know that they do not disperse far from the source. We know that adult numbers are limited by removal. And so basically, just like other large predators, gators are going to be marginalized by urban development. So as urbanization continues in the Southeast, and especially in Florida, over the coming decades, we would expect alligators to be pushed further away. There's still going to be remnant populations. Like, we still have alligators in Jacksonville. They're just not, it's not a very robust population, and recruitment to, adulthood is very limited. So the future research we're doing, we're actually collecting more retention pond data right now because we want to confirm the observed patterns we saw last year. We want to make sure that what we're seeing is consistent over time. We're still tracking St. Johns River alligators because there's ongoing dredging projects in the St. Johns that cause saltwater intrusion. And since we know alligators are sensitive to saltwater, we want to see how that's going to impact. Is it going to push them further upstream over the coming years? The question we really want to get to, though, is how does urbanization affect ecological roles of alligators, right? I mean, we've basically just been talking about presence and absence. We want to know if there's an area that alligators aren't inhabiting anymore, is that, potentially contributing to increases in raccoon populations or rodent populations, right? Prey that are typically targeted by alligators. So with that, I'll thank my funding sources, UNF and the St. Augustine Alligator Farm, my wonderful students who make all this work possible. And this is my contact info if you have any, want to get in touch, and I'm happy to take any questions.
How long has the SNAP program been around, and do you think you're seeing an impact of the 10-foot alligators getting pulled? And so we're going to see a progressive shift to smaller size.
Yeah, so the SNAP program has been around, I believe, since the 90s. I think that's when it started, or maybe the early 2000s, but it's been around for a couple decades at this point. And I think that the impact, the previous impacts were from hunting, right? I mean, hunting was a big impact for many decades before that. And the SNAP program removes from the entire state, they remove and kill about 8,000 alligators a year. So, I mean, I'm not saying the SNAP program doesn't have an impact, but I think the big impact's already happened. And the SNAP program at this point is just sort of maintaining the status quo. Yeah. Yeah.
Like, in the retention ponds, you only found smaller juvenile alligators, right? Do you think that could be because they get like kind of isolated and stuck in these ponds, and then when they grow to adult size, the program gets called on them?
Yeah, that's probably what's happening. The way that their dispersal patterns work is juvenile alligators get pushed out of areas occupied by adults because adults can cannibalize them, right? Cannibalism is very prevalent in crocodilian communities. So they leave those areas and they disperse outwards. And then, as you said, they just get stuck in these areas because it's hard to move from one to another if they want to. And then we've seen it on the UNF campus. We regularly have alligators on the UNF campus, but once they get bigger than 4 feet, people call and they get removed. The other problem is people, of course, right? Because on the UNF campus, students feed the alligators, right? And that's a big public health problem because that's when alligators attack people, is when they've been fed. So, there's a big pond by the cafeteria on our campus. And last summer I was watching this alligator, and every time I walked by, he would approach the boardwalk, right? Because he's expecting people to throw food into the water. So yeah, that's the situation. Yeah.
I think I missed some of your methodology, but when you were doing the counts, how did you tell relative age of the alligators?
Good question. I didn't explain that at all. You can estimate size from an alligator. Actually, it's been scientifically tested. It sounds like it's sort of folklore, but the distance from the eyes to the snout in inches is the length of the alligator in feet. And you can roughly estimate age from size, right? So like a 6 foot long alligator is roughly 7-8 years old. So yeah, we don't know. We don't know the exact ages of these alligators, but we do have a good idea of the size.
How close can you have to get to them before they?
It depends. So the larger alligators are more wary, right? Because they've been around for longer. They've seen humans and they might have been harassed by humans. So it's harder to get close to larger alligators in an urban area. The smaller alligators are not as intelligent or just don't have as much experience, and you can get really close to them, and sometimes you can just grab them out of the water. Don't do that. That's illegal. I don't do that. I'm not saying I do that, but you could do it. Yeah.
So this is slightly off topic, but I would just I guess your opinion with the research that you've done on alligators, since the Southeast is rapidly urbanizing, like you showed in that initial map, and we're experiencing kind of a warming from climate change of the north part of their range. Do you think that those two things are going to kind of work in tandem to help push them more north and to go into states like North Carolina and Southern Virginia quicker than anticipated?
That's a good question. I don't know about quicker than anticipated. It's what you're talking about is definitely happening already. Places like Tennessee are seeing increasing gator numbers, whereas historically, since Europeans got here, right? Historically, they were not that abundant in Tennessee, and Virginia as well. For years, there's been talk about isolated alligator populations making their way into Virginia, or individuals, I should say, but. The areas where they're expanding into are preserves usually. And the areas they're expanding into in Tennessee and Virginia are not heavily populated right now. And I don't think they're expected to become heavily populated. So I don't think they're going to be limited maybe in that way. Yeah, I think it's more they're moving north just because the temperatures are warming up.
Yeah, so I also have the presentation. I might have missed this, but I was curious about, you mentioned the saltwater and saltwater treatment. Are you looking at the proximity to freshwater sources? I know in North Carolina, we'll go from the mainland to the outer banks of Roanoke Island, for the blue crabs and all the food resources out there. So I don't know if that was something that you were looking at in terms of the potential through urban landscape or areas where saltwater might become a barrier for smaller alligators.
Yeah, that's actually what my entire PhD was about. But we saw, we know alligators will go into saltier areas because they have more food available, and then they'll return back to freshwater. That happens in the Everglades, which is where I did my PhD. But in Jacksonville, I don't think that's happening because, one, the access to saltier waters, I don't think actually food is even in higher abundance in the saltier waters in Jacksonville because the St. Johns River has been so heavily impacted by, shipping and pollution over many years. So I don't think they see that advantage when moving into the salty waters. Plus there's so much boat traffic, right? That there are massive container ships that are moving through the St. Johns River every single day, and alligators do not like that. So I don't think they have the incentive to do it. They could do it, but I just don't think they have the motivation.
Maybe as we're transitioning to the next speaker, were you able to hear the plenary?
No, I wasn't, unfortunately, because I was finishing my talk.
I would love to hear. So the plenary had some really interesting thoughts and insight on us humanizing some of these species and in a good way and how that might be helpful. I really wanted to hear your thoughts on doing it with alligators versus cougars, but I'll catch you later.
Okay.
Go ahead.
All right, thank you, Adam.
Thanks.
All right, next, the last presentation in this session is going to be Tom Sankey presenting hidden in plain sight alligator snapping turtle populations within urban waterways of Houston, Texas, Harris County.
Thanks, Anna. So I'm not the primary or the secondary, I'm the tertiary. And you remember Andre was talking about earlier about him not doing any of the work. I really do like even less than he does on his projects. We found out, Aaron and I found out on Friday afternoon that neither Eric nor Mandy could be here because of family emergencies. So apologize for the rockiness of this, but SWCA's tagline is Sound Science Creative Solutions. We're going to do today Sound Science, get up there and wing it. So, okay. Here we go. Forward, backward. How do we work this?
You did a space bar or forward.
There we go. Okay, so AST, it's the largest freshwater turtle in North America. It's the largest hard-shelled turtle in the world. It's sexually dimorphic. The males are typically more, they're larger than the females, and they have a much more bulbous head. It's primarily an ambush predator. It feeds mainly on fish and occasionally on carrion. And the bite pressure is typically 1,000 PSI. There's also some evidence that it might be more than that, up to 1,200. In terms of the range, essentially in the Gulf of Mexico, goes from the Panhandle of Texas to Panhandle of Florida to the Panhandle East Texas and up to Western Illinois. So it occupies a wide range of habitats, including all different types of clear versus suspended sediment, very turbid waters, prefers areas with the key that we're finding is that it requires submerged structure. That seems to be one of the findings of our studies. It's the take-home message here is there's very little movement between watersheds. We're not finding any genetic variability between watersheds or a whole lot of genetic variability within rivers. So the take-home message is it's really an isolated river endemic species is what we're finding. In Houston, it's the fourth metropolitan area. ASTs were purported to be extirpated in as early as the late 1970s. So when SWCA was doing another study in the within about 6 miles from downtown Houston, several alligator snapping turtles were discovered in Buffalo Bayou. So the question was, are there more that are actually going to be found in other parts of the city or of the county? A little bit more information on it is it's federally threatened, or it's proposed for federal threatened status. And the biggest question that if we found one or two as early as 2016, would there be more in the county? So in October 2016, we found several individuals in Buffalo Bayou. It's the farthest southwestern extent. At the time, it was the farthest southwestern extent of the species. Lower right, you can see Buffalo Bayou. It's a typical urban watershed. It is affected by degradation, water quality, suspended sediments, a lot of bank erosion, riparian zone. This is a good example of in the foreground, the riparian zone actually being affected. In the background, it's still intact. The main message here is that our formal study we're very interested in looking at abundance, home range, and habitat utilization within Buffalo Bayou. So this is showing a good bit of, well, so essentially there's quite a bit of variability in the size range. Also in the pop, you can see juvenile, female and male. That's a good indication of the size of the length of the carapace, actually indicating that males in the population, or well, males in general in this species are very much larger than the females. Incidentally, the results of this were published from the 2016-2018 study in the Urban Naturalist manuscript. And this is, let's see. Oh, CPU, right? That's what this is. Yeah. So yeah, catch per unit effort. So there was quite a bit. The mean here is actually between 0.3 and 0.4 per number of turtles per night. So that's actually a pretty decent capture. So we know that the issue here is we know that we're getting good results in where we're putting them in. And at this point, I'm going to turn it over to Aaron to do the rest of the slides and answer all the questions because he's doing most of the work as well.
I've done some of the work, and I've done a lot of the trapping. It's been a blast actually to do it.
Yeah.
And I apologize. Yeah, we just learned that we had to be doing this a couple of days ago. I have my notes here. I'm going to do my best. I think one thing that we definitely wanted to touch on this slide is that we've captured over 120 individually marked turtles in Buffalo Bayou, which is pretty crazy considering 10 years ago they thought that they weren't even there. Like they had no clue that they were there. I guess there were anecdotal things, but it was very exciting for us when we stumbled upon this population and we were able to, get this actually turned into more of like a citizen science thing. It started with SWCA, our environmental consulting firm. We just stumbled onto it. Then we had some, me and Eric Muncher, one of the primary investigators as part of the Turtle Survival Alliance. And we started trapping on our weekends and volunteering and getting all this data. It was really pretty amazing. Yeah, and we just love this project. So in 2021, so in 2021, we actually partnered with the University of Houston and Clear Lake. We actually did a statewide survey of of alligator snapping turtles all across East Texas, but we did expand our search in the Houston Metroplex. And Buffalo Bayou is right here, going along the southern end here. But then we trapped Little Cypress Creek and Cypress Creek and Spring Creek. And then we did one little trapping session on Moose Bayou. But one thing I want to mention here, we have 100+ turtles here, which is the most urban part. And then we go up to Spring Creek and we've trapped a lot up there and we're not seeing many. It's pretty interesting. Yeah, so here's our per unit trap effort. Loose Bayou's our lowest. Spring Creek's pretty low. We did go to Little Cypress Creek, which is a very small creek, and it's amazing. We're almost getting 1-to-1 trap success there. You throw traps in there, you're going to get alligator snapping turtles. It's pretty neat stuff. And then our baseline at 0.3 across the board. So this, I think, shows the size distribution across all different watersheds we've trapped. Kind of the same with the Buffalo Bayou. You can obviously see the males are much bigger. Let's see, and then yeah, let's see. But right here is the juvenile sizes. This kind of shows that we are trapping or catching turtles in every size demographic, which means they're probably healthy populations here in a very urban environment, which is good. We need to ask a lot more questions about that. So 5 years of data from our Harris County trapping. Basically, we've caught 173 individuals from all over Harris County, and our sex ratios are basically 1-to-1, which is also really interesting data. But one thing about this is that we still have many gaps in where we haven't been able to trap. It's an urban environment. There's lots of residential areas we just can't get to. One interesting thing about it is when we're trapping in Buffalo Bayou, we ran across some kids fishing, and they were fishing for alligator gar. They were doing their YouTube thing, and they were catching big alligator snappers. And so we were able to get in touch with them, and they took us to spots where they had seen big alligator snapping turtles, and so we kept finding more pockets of these alligator snapper populations within Buffalo Bayou. It just shows that we need to trap more and look at more of these areas. We did do a telemetry study on these guys for about 2 years, found out that they don't move very much at all. Maybe, I think the largest movement was maybe a quarter mile, but that was one individual. The rest, 100 meters tops. They just stay put. So we think when these pockets we haven't trapped in, there could be many more turtles there. Another thing real quick, right before our survey started, there was no growth curves for the species in Texas. So with the 5 years of data that we've collected, we were able to develop this growth curve that shows, some growth over time of males and females. And we have 3.5 more years of this study that we want to do, maybe more. So after 10 years, we'll redo this growth curve and see what we can get. We found that this is probably the densest population of alligator snapping turtles in Texas. It's pretty amazing. It's in downtown Houston. We've seen all different sizes of animals. This is actually an image of, this is the only female that we've ever seen nesting, climbing up on a bank and nest. And this bank was at I don't know what the degree was. I don't know how she got up there, but and then also this was the closest to downtown that we've seen this nest, which is pretty neat. We have seen hatchlings in all of the sizes, but another take-home point is that, why are these guys doing so well in Buffalo Bayou, in the middle of the city? Like, are they protected from poaching? Are they just congregated down there? A lot of the bayous going into the river, into Buffalo Bayou right now, are these trapezoidal concrete streams that offer zero habitat. So they just congregated in this tiny little highly densely populated area, but they seem to be doing fine. Their weights are doing fine. They're breeding. So it raises some interesting questions. Continued research. We want to do at least 10 years of trapping here. We want to continue doing telemetry. We're actually got a grant to do some satellite trackers on them, which should be interesting. We especially want to know where the females are coming up to the nest. I think I'm running out of time, so.
Yeah, 2 minutes.
Okay. We want to find out where the nesting habitat is, and maybe we can find where nesting habitat, since it is proposed to be listed, we can maybe protect some of those areas as, a conservation effort to keep them safe. And I just want to say thanks, especially to the Turtle Survival Alliance, Houston Parks and Recreation, Texas Parks and Wildlife, Kelly Nord, who was on here. He's actually here today. I think he's actually talking in another talk, but he's been a great asset to helping us out with the Houston stuff. And from there, any questions?
Two curiosity questions. What type of trap are you using to catch them? And then does this habitat also support soft shells and common snappers? Are there turtles
Yeah, yeah, good question. We use 3 foot to 4 foot hoop nets. And we do get soft shells. They're the worst to get out of nets. And yeah, and we do see some sliders. We have caught map turtles in our traps too. We do get a lot of other bycatch like alligator gar and stuff like that. And I'm sorry, what was your other question?
I think you covered them all.
Okay. Yes.
Are you guys hoping to expand into doing like genetic testing on them to maybe help
Yes.
your case that, you know, even if there's a lot of them, like
Yes.
maybe they're not genetically diverse?
Yeah, so when University of Houston came and partnered with us, we did a lot of blood work, and so we have in tissue. They took a lot of tissue from the animals as well. That's still kind of a work in progress, but yeah, we'd like to keep doing that as well as much as we can, especially as we move up to the other watersheds. Even in Houston, are these guys the same? Do they move 5 miles, and how do they get there? Because right now we haven't found them. They don't go to other watersheds.
I was just wondering, given your success finding new locations for those kids that were fishing, have you considered exploring any other like citizen science reporting tools or anything like that?
In fact, we did that. University of Houston. We published a paper. Didn't Mandy publish a paper?
Mandy Gordon published that paper. That was last year, the year before, about that. I don't recall any of the results. One of the things that we think, and I believe it was discussed in that one. I know that Eric Muncher, the primary, and I've talked quite a bit about, we believe that, the question is, in the middle of urban, why aren't they there? And we think that critical habitat really is the nesting areas. And if you know anything about Houston, it's almost all privately owned. There's the Memorial Park that's there. That is public down to the bayou, but almost all the remaining 28 miles, 29 miles of riparian zone, the owners own to the, and they're very particular about people coming up and down. And there's also most of the riparian zone is not developed and hasn't been in the areas where it has been. So one of the things we're interested in doing that Mandy and Eric and Aaron and I have talked about is actually getting outreach, but we also think that the less people know, that the better they're doing. So there's that question.
Yeah, I mean, you mentioned poaching, and so I'm not familiar with that's a big then problem.
The poaching problem has been a significant part in the almost extirpation and in the huge reduction in Louisiana. And still to this day, you were talking the other day about they still have a 1 turtle per day.
I think so. We could still
take 1 turtle per day. So we think that that's one of the things that has reduced. There's not that much in Texas. It's actually imperiled in the state, and I believe it's protected now. It has some state protection, but the reason why you have such a reduction in Louisiana is directly attributable. There's a lot of anecdotal information about that. It's directly attributable to poaching in the state.
Yeah, Louisiana, they've collected so many of their animals that they're now coming into Texas to collect animals.
Yeah, they're
to poach.
Our brethren from the east. I hope nobody's from Louisiana in here. Sorry. Yes.
Was it Hurricane Henry?
Harvey?
Harvey
That flooded Houston. Did that happen during your study collection?
Yes,
And then
that's
did that
very
have effect on?
That's very interesting because that happened right after we started. Harvey was in 2017, so we had already been trapping, and in fact, one of our turtles ended up on top of one of the bridges that went over Buffalo Bayou. That's how bad though, and so that turtle's now known as Harvey. But it's interesting, Houston, the Buffalo Bayou, it's concrete, so it's actually makes it very hard for trapping. We can't trap if it's even going to be a chance of rain. The water raises like crazy, but it's amazing how this water will just flush through, and these turtles are just still there. What are they? They just
stay,
yeah.
Not
It's pretty cool.
Not 100%, but there's very little movement up or down, or as we've seen also across watersheds. That's one of the reasons with the genetic, they can be trapped out. They can be extricated very quickly because they're very yeah.
As a naive Californian, what are people doing with 1 turtle a day? I don't understand why somebody
they're eating them.
They're eating them
Okay,
still.
okay. They make soup out of it. That's
Yes.
very culturally significant.
Yes.
Very significant.
Yep.
I have a question. So like you were saying like they're in these creeks that are surrounded by concrete. So it's like primarily runoff water from the roadway that's going into these creeks?
Yeah, so
Yeah.
Buffalo Bayou's
Go ahead.
kind of natural, yeah, but some of the bayous that where they used to be historically found, they've been just channelized and concrete lined.
This-
So
Yeah, so Buffalo Bayou is the outfall of the Barker Reservoir. It's actually a flood control reservoir. So, and almost all of the flows are stormwater flows, but it's also the release from there, stormwater release. So there's quite a bit of areas that it's just a stormwater conveyance until it gets down to downtown Houston. That's about I think it's 24 some miles. So part of it has actually been channelized, but there's a good part that's not.
So has there been any like water, water samples where you collect these hurdles, the water quality is that they're looking at? Because then like people that consume them that might be like dangerous for their health. If there's like accumulation of like some type of chemicals in runoff water.
yeah, I mean, I know that the city of Houston, I'm sure, does water testing all through there, but right now with our funding, and we cannot do that now. It would be interesting to do that now, to even test some, collect turtles and see if there's any accumulation in tissue.
So we met this morning. You're from upstate New York,
Yeah.
correct? So do you all want to come to Texas to do a study like that?
Sure.
Absolutely.
There you
Harris
go.
County Flood Control District would be the one to petition for that.
All right. Okay.
Thank you so much.
Have a coffee break downstairs. Help yourself. I'm gonna change out.
Thanks, man. Sorry, I like took over for you. I apologize. That's why I was like, oh, let him do his job. But I'm gonna change his computer out because it's too slow.
And actually, I am in the next session and I wondered if you wanted my thing now or come back in a bit. to give it to you.
If you.